right, so how do we understand this kiss and run phenomena? There is still some controversy surrounding kiss and run, but most people accept the idea that it does exist. There's only a few people that still argue against its existence. And we know it from various kinds of experiments that place a fluorescent dye or substance or particle inside synaptic vesicles. And one has to think about how to load those into vesicles and then how to release them from vesicles. Once loaded into vesicles, this is a diagram of an experiment done with something called a quantum dot, which is a fluorescent particle which is made out of semiconductor material. But they're about 15 nanometers in diameter, so they'll fit inside a synaptic vesicle. And once inside, and once the vesicle is acidified, because synaptic vesicles are pH 5.5, whereas the cytosol is pH 7.4. So there's a pH difference there. And the dye is pH sensitive, so it will be quenched when the vesicle is acidified, and it will be fluorescent when the vesicle is exposed to the cytosol. So you can see that 15% difference in the fluorescence emission curves based upon the pH of the environment. So if one were to focus a very high-powered microscope at a synaptic vesicle that had the quantum dot in it, and here we can see that we have quantum dots at two places in this image, and the experimenters Zhang and Chen have also labeled all of the synaptic vesicles with the dye FM464. So they have 4 FM464 labeled vesicles, and 2 of those have quantum dots in them. So you can see that they have verified that the quantum dots do get into the vesicles. Then if they look at the wavelength associated with the quantum dots as opposed to the FM464, when a vesicle kisses, so it opened its fusion pore but has not let the quantum dot out, but the quantum dot goes from pH 5 to pH 7.4, it all of a sudden turns on and emits fluorescence. As that vesicle closes, that fluorescence decays with the time constant equivalent to the acidification process. However, if the vesicle went through full collapse and fusion and released the quantum dot to the cytosol, or not to the cytosol, to the extracellular space, where it could be washed away by the flowing solution, because all of these experiments are done in a tissue culture environment where one can actually focus very carefully and get very close to the presynaptic nerve terminal, then the fluorescence essentially goes away with the speed of the solution flow, taking that quantum dot out of the focal area. So one can distinguish small kiss and run events from large full collapse and fusion events. And just by then quantifying the amount of fluorescence involved in these two different events and plotting fluorescence intensity versus a histogram of the number of events, one can see in the closed state all of the time spent right around no change in fluorescence. You can see the number of events that would represent a kiss and run change in fluorescence intensity, and you can see a histogram of the number of events that would represent a full collapse and fusion event. So Zhang and Chen use this to map multiple evoked responses. If they stimulate very slow rate once every 10 seconds for a full 2 minutes, which is basically 12 stimuli, and then they look at all of the traces they get, so traces which there's no response until finally a late full collapse and fusion, traces with an immediate kiss and run event followed by a full collapse and fusion event, an immediate full collapse of fusion, etc. So each line in this bar graph represents one sweep or one evoked response, and the red are kiss and run events, the blue are full collapse of fusion events, and you can see the time in between when a vesicle might undergo both. Oh, kind of an interesting idea. And sometimes even a couple, but not here. So they look at all of these accumulated events, and they look at the overall incidence of the number of events of each kind they get per stimulus, because remember, there's only 12 stimuli here. So in some of these, it took multiple stimuli before you got to a full collapse and fusion, or before you get to a kiss and run. And notice the time scale. It takes a while. Under normal conditions, at this slow rate of stimulation, they see many kiss and run events to the first stimulus, and that number of events falls off by about the fourth or fifth stimulus to a fairly steady state, very few, low number. Full collapse and fusion, however, the number of full collapse and fusion events increases with the number of stimuli, or with the history, the recent history of activity at that particular synapse. So this gives us the idea that at any particular synaptic site, presynaptic site, whether a choice is to recycle via kiss and run or the recycle via full collapse and fusion may be dependent on the history of activity at that site. It also may depend upon which vesicle pool is available. In the next experiment, they use a high concentration of sucrose. It's called hypertonic sucrose, so osmotically large amounts of sucrose, which has been known for a number of decades now, that causes fusion of all of the vesicles that are docked and primed and ready to go. So we call those vesicles which are sitting there waiting for a stimulus the readily releasable pool. And high concentrations of sucrose for short periods of time, on the order of seconds, will osmotically force release of the contents from the readily releasable pool. After that readily releasable pool is depleted, then if this very slow once every 10-second stimulation is given, the incidence of kiss and run is very, very low, and the incidence of full collapse and fusion is fairly stable until even then the pool may be depleted. So not only is the choice between kiss and run and full collapse and fusion dependent upon the previous history of recent use of that synapse, but also on which pools are available. So it appears that the readily releasable pool may be the pool that is undergoing the kiss and run, because in the absence of the readily releasable pool, we see very low incidence of kiss and run. Now the bottom graphs compute the ratio of the kiss and run number per stimulus to the full collapse and fusion number. So initially, again, full collapse and fusion is not favored, kiss and run is favored, but that drops off with stimulus number. And you can see the difference here, which is the same computation for the sucrose experiment. But what about changing the frequency of stimulation? In the next experiment, Zhang ups the stimulation frequency to 10 hertz. So quite a large number of stimuli now in this 10-minute period. And here they see sometimes multiple kiss and run events, 1, 2, 3 kiss and run events with an interval time between it before an interval that leads to full collapse and fusion. And again, they look at multiple events under this kind of faster stimulation, and they plot the same kiss and run ratio. And here, there's many, many more kiss and run events than on the previous graph, which is now the lighter orange squares, which have just been regraphed here. So at a higher frequency, kiss and run is favored. And eventually, with many more stimuli, full collapse and fusion becomes higher probability than the kiss and run, because we're below the 0.5 line here.